An image unlike any other has fueled decades of investigation, controversy, and debate. The Shroud of Turin stands as one of history's most enduring enigmas, a relic that continues to challenge both faith and science. It is a burial cloth bearing the faint imprint of a man many believe to be Jesus Christ after crucifixion. For centuries, arguments have raged over its origin, but today, the shroud has returned to the spotlight in a profound and unexpected way. Deep within the ancient linen fibers, scientists uncovered something extraordinary: DNA. When researchers began analyzing it, the results left them stunned. For millions of believers, the shroud represents the burial cloth of Jesus Christ, the only physical remnant of the resurrection, a silent testimony, a fifth gospel written not with ink, but with blood. Skeptics, however, tell a very different story. To them, the shroud is the most elaborate and convincing medieval forgery ever produced. Some have even suggested that Leonardo da Vinci himself may have created it. Others argue it was the work of an unknown master, intentionally crafted to deceive pilgrims longing for miracles. For more than 6 centuries, the divide has remained unbroken. Faith on one side, logic on the other, and neither willing to yield. But the modern era changed everything. The age of philosophy and speculation gave way to something far more unforgiving. Science entered the debate, cold, methodical, and relentless. Genetics, high-energy physics, spectroscopy, and forensic analysis opened doors once thought impossible. Researchers no longer viewed the shroud as a sacred object alone. They approached it as a crime scene, as evidence in a mystery unsolved for 2,000 years. A biological archive silently preserving history. DNA was extracted and sequenced from the fibers. Every thread was scanned with X-rays. Blood stains were broken down into molecules, atoms, and elemental signatures. Scientists expected straightforward answers, traces of a single person, or perhaps remnants of pigments from a medieval artist. What they found instead was anything but simple. Geneticists at the University of Padua, alongside physicists working within the sealed laboratories of ENEA, uncovered data that shook even the most seasoned experts. The results didn't merely challenge existing explanations, they dismantled them. Both traditional religious interpretations and skeptical theories collapsed under the weight of the evidence. The shroud ceased to be just cloth. It became a map, one that traced a journey beginning 2,000 years ago. It recorded suffering with a precision that defies replication, and it revealed evidence of an intense burst of energy, one that modern physics still cannot fully explain. This marked the beginning of the most comprehensive forensic investigation ever undertaken. Invisible trails would be followed, microscopic pollen grains, molecular residues, biological markers. Step by step, researchers sought to uncover whose blood truly soaked into the linen, and why it speaks so clearly of torture, expressed not through words, but through biochemistry itself. Scientists also turned their focus to the image formation, an image that behaves simultaneously as a photographic negative, a hologram, and an X-ray. One question in particular stunned the scientific community. Why did DNA analysis force researchers to conclude that this artifact could not have originated in Europe, and that it bears witness to events that reshaped human history? To understand the shock of modern discoveries, we must return to the beginning of scientific shroud research, modern syndology, to the moment humanity first truly saw the face hidden within the linen. That moment came on May 28, 1898, in Turin. A lawyer, city councilman, and amateur photographer named Secondo Pia was granted rare permission by King Umberto I to photograph the relic during a public exhibition held to celebrate the prince's wedding. Photography at the time was a demanding process, slow, technical, and unforgiving. Pia hauled a camera the size of a suitcase onto towering scaffolding inside the cathedral. He used powerful magnesium flashes to illuminate the dim interior. 2 large glass plates, each measuring 50 by 60 centimeters, were exposed. Late that night, alone in his darkroom and guided only by the faint glow of a red lamp, Pia immersed one of the plates into developing chemicals. And then, something extraordinary happened. As the image began to appear, Pia nearly dropped the priceless negative. What emerged seemed impossible. On the photographic negative, where light should become dark and dark should become light, there appeared not a faint or distorted stain, but a sharply detailed, high-contrast positive image. A face emerged, deep-set eyes, closed eyelids, a broken nose, a mustache and forked beard, bruising on the right cheek. For the first time, the figure on the shroud was no longer a vague shadow. It was a man. The expression was striking. Calm, authoritative, almost regal, yet unmistakably belonging to someone who had endured extreme physical torment. The impact was overwhelming. This was not merely a surprise. It was a turning point. Ordinary drawings or paintings, whether made with pigment, charcoal, or blood, behave predictably when photographed. Light becomes dark. Dark becomes light. The result appears distorted and unnatural. A flattened, mask-like image. The shroud behaved differently. When photographed as a negative, it appeared as a true black and white photograph of a real human being. That single fact changed everything and remains impossible to ignore, regardless of how determined one may be to dismiss it. What the human eye sees on the cloth is not a normal image. It is already a negative. And that raises a profound question. Who in the Middle Ages understood the principles of photography? Who knew that light could be reversed to produce a hidden image? The answer is simple. No one. Photography would not exist for another 8 centuries. The human brain cannot naturally perceive the world in negative, let alone recreate it with such precise gradations of light and shadow. Who could intentionally produce a perfect negative image without any way to preview or confirm the result? This was the first true mystery, the first fracture in the wall of skepticism. The shroud did not behave like a painting. It behaved like a photographic plate, capturing a single instant in time. Over the years, the cloth has been examined using X-rays, ultraviolet light, infrared imaging, and laser scanning, each revealing new layers of a mystery that refuses to be solved. Yet the greatest secret was never hidden in the image itself. It lay in the dirt, the dust, the microscopic particles trapped between the linen fibers through 2,000 years of movement, handling, and exposure. In 2015, a team of geneticists and biologists led by Professor Gianni Barcaccia of the University of Padua was granted unprecedented access to the relic. Their objective was as ambitious as it was unsettling: to locate and analyze DNA. They were not searching for the DNA of God. Science has no concept of what such a signature would look like. There is no reference sample, no benchmark for comparison. Instead, the researchers sought something different. They aimed to reconstruct the history of the cloth itself, where it had traveled, who had touched it, and whose hands had held it across centuries. To accomplish this, they employed specially engineered sterile micro-vacuum devices fitted with ultrafine filters. These instruments gently extracted microscopic particles of dust, pollen, and organic material, not only from the surface of the linen, but from the deep spaces between the warp and weft threads, where ancient traces can remain sealed for millennia. They also examined particles collected during the 1988 restoration, which had been carefully preserved in archival conditions. This work pushed the limits of what science could safely attempt. Even the smallest modern contamination, a sneeze, a breath, a single flake of skin from a priest, conservator, or technician, could overwrite ancient genetic signals with modern noise. The samples were transported to ultra-clean, controlled laboratory environments. There, sequencing began using next-generation sequencing, or NGS. The scientists focused specifically on mitochondrial DNA from both plants and humans, and for a critical reason. Unlike nuclear DNA, mitochondrial DNA exists in hundreds of copies per cell, is inherited exclusively through the maternal line, and survives far longer in ancient materials. It serves as a powerful and reliable marker for geographic origin and patterns of human migration. For weeks, computers ran continuously, decoding millions of nucleotide sequences and comparing them against global genomic databases representing populations from across the world. When the final visualizations appeared on the screens, maps of genetic haplogroups and ancestral lineages, the researchers realized they were seeing something that defied any simple explanation or possibility of forgery. This was not the genetic profile of a single individual. It was a genetic portrait of humanity itself. The findings were published in the highly respected journal Nature Scientific Reports, and the reaction was immediate and explosive. Shock waves rippled through the scientific community. Researchers had expected to find one dominant genetic signature. If the shroud were a medieval forgery created in France, as skeptics have long argued, then European DNA, particularly French or Italian, should have overwhelmingly dominated the results. If, on the other hand, the cloth were an authentic relic from Jerusalem that had never left the region, the genetic traces should have pointed almost exclusively to the Middle East. That is not what the data revealed. What emerged instead was complexity bordering on chaos, the entire world encoded into a single piece of cloth. The shroud carried genetic fingerprints from populations across vast regions of Eurasia and Africa. First, the Middle East. Scientists identified haplogroups commonly associated with the Druze, a tightly knit and historically isolated ethno-religious community inhabiting mountainous regions of Israel, Jordan, Lebanon, and Syria. Druze DNA is considered extremely ancient and remarkably stable, having changed little over thousands of years due to prolonged isolation. This made it a powerful marker of geographic origin and strong evidence of a Middle Eastern connection. Second, Western Europe. Haplogroups such as U5b and H1 through H3 appeared exactly as expected. Since the 15th century, the shroud has been housed in Europe, first in Chambéry, then in Turin. It was handled by the Poor Clare nuns who repaired it, by members of the House of Savoy who owned it, and by countless European pilgrims over the centuries. Third, North and East Africa. Haplogroup L3CM1A1 emerged, pointing to regions such as Egypt and Ethiopia. This was unexpected and deeply intriguing, suggesting contact with some of the earliest Christian communities on the African continent. Fourth, South Asia. Haplogroups M39, M56, and R8 appeared, genetic markers characteristic of the Indian subcontinent. And most astonishing of all were traces from East Asia. Haplogroups such as D4 and G2A, markers commonly associated with populations in China, were present. China, India, Africa, Europe, the Middle East, all encoded within one ancient cloth. How could this be possible? If the shroud were a forgery created in the damp basement of a French abbey around the year 1350, how could traces of Chinese and Indian DNA possibly exist on it? In the medieval world, globalization did not exist. Long-distance contact occurred, yes, but not at a scale sufficient to leave such widespread, clearly identifiable genetic signatures on a single piece of fabric. The answer turned out to be even more remarkable than the mystery itself. The shroud is not merely a burial cloth. It is a traveler. When scientists in Padua mapped the genetic data, they made a stunning discovery. The distribution of genetic markers aligned with remarkable geographic precision along an ancient historical route that many scholars had long dismissed as legend, the journey of the man-made image known as the Mandalion. According to early Byzantine, Syrian, and Arabic sources, the shroud was folded in such a way that only the face was visible and displayed within a special frame. This folded relic, known as the tetradiplon, did not remain in Europe. It moved across the ancient world. The journey began in Jerusalem, the site of death and resurrection. From there, it traveled to Edessa in the 2nd century, where it remained for centuries, hidden within the city walls before being rediscovered. Edessa stood at a vital crossroads of the ancient world, the Silk Road. Caravans from China, India, Persia, and Arabia passed through the city, carrying silk, spices, and goods. Merchants, pilgrims, and diplomats all came to venerate the relic believed to protect the city. They breathed near it. They kissed its casing. They touched the reliquary. With each encounter, microscopic traces, skin cells, hair fragments, sweat settled onto the cloth. Layer upon layer, century after century, the DNA of the world accumulated on its surface like invisible dust. The journey continued. Next came Constantinople. From 944 to 1204, the relic was held at the heart of the Byzantine Empire. The emperors of Byzantium acquired it by negotiation or by force and brought it to what was then the greatest city on earth. Constantinople was a true megacity of the ancient world, a place where people of every race, culture, and region converged. Then, in 1204, during the chaos of the Fourth Crusade, the city was violently looted. In the aftermath, the shroud vanished. Its next stop was Athens and Greece around 1205. After the fall of Constantinople, the relic passed into the hands of French knights and was kept in Athens for a time. Then came France, around 1353. This moment marks the shroud's first clearly documented appearance in Western Europe, in the possession of the knight Geoffroy de Charny. And it is here that the genetic evidence delivers a devastating blow to the medieval forgery theory. A forger working in 15th century Europe could not possibly have collected dust and DNA from people originating in China, India, the Middle East, and Africa, regions known only through distant travel accounts such as those of Marco Polo. Nor could such a person have intentionally embedded those traces into a cloth in a way that would deceive genetic scientists 6 centuries later. The DNA found on the shroud is not random contamination. It is the collective biological memory of humanity. Dust from sandals, garments, and hands of countless individuals who encountered this face over thousands of years. This biological record demonstrates the antiquity of the cloth and its Eastern origin more convincingly than any radiocarbon test ever could. But human DNA tells only part of the story. The other half is written in plants. Palynology, the study of pollen, provided another crucial piece of evidence. Two highly respected experts approached the problem independently. Professor Avinoam Danin, a renowned Israeli botanist from the Hebrew University of Jerusalem, and the Swiss forensic scientist Max Frey, one of the pioneers of forensic pollen analysis. Working separately, both examined pollen grains trapped deep within the linen fibers. Using adhesive tapes, they carefully lifted microscopic samples from the cloth. What they discovered sent shock waves through the botanical community. Pollen from 58 different plant species was identified on the shroud. 17 of those species are native to Europe, exactly what would be expected given the shroud's documented presence there over the past 6 centuries. But the majority originated elsewhere, plants native to the Middle East, Turkey, and the Anatolian steppe, perfectly matching the ancient route through Edessa and Constantinople. Then came the most striking discovery: a group of plants that grow nowhere else on Earth, species found only within a narrow geographic corridor between Jerusalem and Jericho. At the center of this finding was one plant in particular, Gundelia tourneforti, a thorny desert shrub, a type of thistle. Its pollen accounted for nearly half of all samples identified on the cloth. An enormous and highly unusual concentration. Why would pollen from a thorny desert plant dominate the fabric of a burial cloth? Why would thorns be associated with a man treated as a king? The answer emerged when historians and theologians revisited descriptions of Christ's passion, the crown of thorns. Gundelia tourneforti, with its long, rigid, needle-like thorns, is precisely the type of plant Roman soldiers could have twisted together into a mocking crown for the King of the Jews. Suddenly, the pollen began to speak. This plant blooms around Jerusalem during a very specific time of year, early spring, coinciding with the Jewish festival of Passover Pesach. The discovery of an unusually dense concentration of this pollen, particularly around the head and shoulder regions of the cloth, serves as direct evidence that the body was crowned with it. A second botanical clue came from Zygophyllum dumosum, a plant endemic to a very limited region, growing only in the Judean desert and parts of the Sinai Peninsula. Its pollen was also found on the fabric in significant quantities. No medieval forger working in France could have obtained pollen from plants native exclusively to Israel and then applied it invisibly at a microscopic level to a piece of cloth. A forger would have used paint. Pollen cannot be painted. It acts as an invisible seal from the scene itself. A geographic fingerprint that cannot be forged. For many years, particularly during the highly rational 19th century, skeptics claimed the reddish stains on the shroud were nothing more than pigments, ochre, cinnabar, or tempera mixed with gelatin. That argument collapsed in 2017. A team of Italian researchers led by Professor Giulio Fanti of the University of Padua, working alongside physicians from a hospital in Trieste, examined the stains using transmission electron microscopy and Raman spectroscopy. At the nanoscale, they found no pigment. They found blood, human blood. Type AB, one of the rarest blood groups, yet one that appears frequently on ancient Christian relics, including the Sudarium of Oviedo. But this was not the blood of a healthy individual. Scientists identified nanoparticles of creatinine and ferritin bound to hemoglobin. Such extreme concentrations occur only under one condition: severe fatal trauma. Prolonged torture accompanied by dehydration and massive muscle damage. When muscle tissue is destroyed by repeated blows, a process known as rhabdomyolysis, creatinine floods the bloodstream in enormous quantities. And the blood on the shroud recorded exactly that. This is a biochemical scream of pain. The analysis revealed that the man wrapped in the cloth did not simply die. He was beaten to a state already incompatible with life before crucifixion even began. This aligns precisely with the gospel accounts of Roman scourging carried out with flagella, leather whips embedded with metal weights. The body bears evidence of more than 100 blows. The chemistry of the blood tells a story of suffering no paintbrush could ever imitate. An artist can paint the appearance of a wound. No artist can reproduce the biochemical signature of extreme trauma, polytrauma, kidney failure, and hypovolemic shock. Another long-standing mystery concerned the color of the blood. The stains appear red. Normally, ancient blood darkens to brown or black over time. Scientific analysis revealed unusually high levels of bilirubin, a substance released by the liver during extreme stress and severe trauma. Bilirubin preserves the red coloration of blood for centuries. The blood of a tortured man remains red. This is not mysticism. It is biochemistry under extreme stress. At this point, many ask the same question. What about the famous radiocarbon dating? In 1988, radiocarbon testing appeared to resolve the debate once and for all. 3 of the world's leading laboratories, Oxford, Zurich, and Arizona, announced their results with 95% confidence. According to their measurements, the fabric dated between 1260 and 1390, the Middle Ages. A forgery. The world accepted the verdict. Even the church stepped back, referring to the shroud as an icon rather than a relic. But science does not stand still. 3 decades later, researchers identified where the fatal error occurred. The mistake was not in the technology. It was in the human decision of which sample to test. In 1988, a tiny piece of cloth no larger than a postage stamp was cut from the very edge of the shroud. That corner had been handled countless times over centuries, grasped by bishops and cardinals during public displays. Sweat, skin oils, candle wax, bacteria. It was the most contaminated part of the cloth. Further research by chemist Ray Rogers of Los Alamos National Laboratory revealed something even more damaging. That corner had been expertly repaired during the Middle Ages, so skillfully that the repair went unnoticed. The sample tested was not representative of the shroud as a whole. With that realization, the certainty of 1988 began to unravel. The medieval restorers did not use modern, invisible mending. Instead, they reinforced the damaged edge by weaving new cotton threads into the fabric, carefully dyed to match the aged linen. Later chemical analysis of the very sample tested in 1988 confirmed the presence of cotton fibers. That detail alone raised an immediate red flag. The main body of the shroud contains no cotton whatsoever. It is woven entirely from linen. To conceal the medieval repair, cotton threads were dyed with alizarin to match the aged fabric, and gum arabic was used as a binding agent. In other words, the material tested in 1988 was not part of the original cloth. The laboratories did not date the shroud itself. They dated the patch. What they measured was medieval cotton combined with centuries of accumulated grime, not first-century linen. This was not a minor technical issue. It was a profound sampling failure and a clear violation of fundamental archaeological protocol. Attempting to date the most contaminated and heavily repaired section of an artifact is like trying to determine the age of an ancient statue by analyzing the chewing gum stuck to its base. Once this flaw was recognized, scientists began searching for a method capable of bypassing contamination altogether. In 2022, Italian physicist Liberato De Caro of the Institute of Crystallography in Bari introduced a fundamentally new approach, one that does not rely on organic residue at all. The technique is known as wide-angle X-ray scattering, or WAXS, and it transformed the investigation. Rather than examining contaminants, this method analyzes the aging of the linen itself, specifically the cellulose within its fibers at the atomic level. Over time, linen undergoes predictable changes. The long polymer chains of cellulose gradually break down. Its crystalline structure deteriorates under constant exposure to background radiation, humidity, and temperature. This process functions as the fabric's internal clock. De Caro compared measurements from the shroud to linen samples of known age. From textiles used to wrap Egyptian mummies dating back to 3000 BC to medieval fabrics from the 10th through 14th centuries. The results stunned researchers. The aging curve of the shroud's cellulose did not match medieval linen at all. It was significantly older. In fact, the molecular structure of the shroud's linen aligned almost perfectly, with remarkable precision, to linen fragments recovered from the fortress of Masada in Israel. Masada fell in 74 AD, and the textiles found there are securely dated between 50 and 74 AD, the 1st century, the era of Christ. Using advanced X-ray techniques, scientists effectively pushed the origin of the Turin shroud back more than 1,000 years, placing it squarely within the heart of biblical history. In 1988, science appeared to extinguish the miracle. In 2022, with more refined tools, it reignited it. Yet, one question still remains. How did the image form? There are no brush strokes, no pigments, no ink. The image exists only on the surface of the linen, no more than 200 nanometers deep, hundreds of times thinner than a human hair. Scrape it, and it disappears. This is not paint. It is a chemical alteration caused by oxidation and dehydration. A scorch without fire, a trace left by an unknown source of energy. Scientists attempted every known method: acids, heat, radiation. Only one process produced a comparable effect: a short, intense burst of vacuum ultraviolet radiation. But such lasers did not exist in antiquity. To imprint the image across 4 square meters of fabric, the body would have had to emit an unimaginable pulse of energy lasting less than a billionth of a second, powerful enough to encode the image, yet gentle enough not to destroy the cloth. In 1976, NASA imaging technology revealed something even more astonishing. The shroud contains a perfectly accurate 3-dimensional image. The intensity of the imprint corresponds precisely to the distance between the body and the fabric, something no artist, ancient or modern, could ever reproduce. Even more extraordinary, digital analysis revealed faint impressions of coins placed over the eyes, matching rare lepta minted under Pontius Pilate around 29 AD. The probability that a medieval forger could have known this is effectively zero. The shroud also bears dust from Jerusalem, pollen from plants that bloom only near the city in spring, and blood containing biochemical markers of extreme trauma. The nails pierce the wrists rather than the palms, displaying precise forensic accuracy, including nerve-induced thumb retraction, layer by layer, discipline by discipline, biology, chemistry, physics, geology, history, numismatics. The evidence converges on a single place and time: Jerusalem between 30 and 33 AD. The shroud resists explanation. It is not a painting. It is a holographic imprint of a real human being. A forensic record, a silent witness to a moment that altered history. Returned to its vault, the shroud remains mute. But the data speaks. Legend, faith, and belief are now reinforced by measurable scientific evidence. Perhaps one day, humanity will understand the burst of energy that left this trace. Until then, it stands as a fifth gospel, an eloquent testimony to a world-changing event. Do you believe science will ever fully explain a miracle? Or are some doors meant to remain open, not to reason alone, but to the heart